Speaker this afternoon, Linda Petzold. She has a very outstanding, distinguished record in computational science and engineering. She is a member of the National Academy of Engineering, her own department. I guess you're in both computer science and in engineering. And she runs a computational science and engineering program at the University of California, Santa Barbara. She is the winner of many prizes and distinctions. She won the AWM Siam Kowalewski Prize, the Dahlquist Prize for numerical solution of differential equations, and the first Wilkinson Prize for scientific software. A record of service to the community in the United States especially has been absolutely extraordinary. It goes on for pages and pages on the web. I saw recently that she lectured to young female academics especially about how to say no. But it's a talent that she has never developed herself. This is her third time in Australia in the last 2 years, and we are very pleased that this time she said yes. Please welcome Linda to speak about discrete stochastic simulation of spatially inhomogeneous biochemical systems.
Thanks, Ian. I'd like to thank the organizers for bringing me here to speak at this historic first meeting of the Pacific Rim Mathematics Association. Is that right? Yeah, I think it's a great idea, Pacific Rim. Of course, that's not my idea, right? But anyways, yeah, there's a little bit of a story on that talk about how to say no. I got this email from the AWM about a Siam meeting that I was planning to go to anyway, and they said, can you give a talk about, I forget what it was, challenges to overcome or what you learned in all your years of being an applied mathematician and things like that. And at first I was going to say no, and then I thought, wait a minute, I have this huge problem, which is too many things to do, and this huge problem with saying no, and I thought, okay, well maybe this is my excuse to learn how to do it. So I went to everybody and said, how do you do it? And I came up with all these ideas about how to say no, and I've even got a list and an email folder of things that I've probably said no to, but there's still this problem. Okay, so today I'm going to be talking about discrete stochastic simulation of spatially inhomogeneous biochemical systems. Much of the information from this talk up until the last third is available on my website, www.engineering.ucsb.edu/CSE for computational science and engineering. Okay, so this talk is really centered in a subject that I've been working in for at least the past 8 years, which is systems biology. And systems biology has been in the news a lot lately. There's a famous article from the Wall Street Journal about biologists' new approach, do not shoot the radio. And the idea is that how would biologists learn to fix a radio if they knew nothing about it? Well, they did a large grant, and they purchased hundreds, probably more like tens of thousands of identical working radios. Okay, then they take a big shotgun and shoot away. Okay, and then there would be some components hanging around. Okay, and then they'd study those components and learn a great deal about them. But chances are they'd still not have a working radio. How would an engineer, electrical engineer, fix a radio? They'd probably take a single working radio and attach an oscilloscope to it and try to understand how the different components are working together. Maybe not knowing what the components each of the components are doing in a great amount of detail. And so systems biology is about fixing an engineer's approach to fixing the radio, the biological radio. I've got too many switches here. And systems biology has been a lot in the news lately. It's a little bit worrisome. I don't like to work on such a hot topic. Too much pressure. Even Harvard University Medical School launched a new department of systems biology. Okay, so much for that. What is systems biology? I get this question all the time at UCSB. The U.S. government funded a working group to go around the world and try to answer that question, and of course how we, are doing in systems biology compared to everyone else. This is American way, right? Okay, so they came up with this definition that it's the understanding of biological network behavior through the application of modeling and simulation. That's where I come in. I'm a modeling and simulation person, tightly linked to experiment. And so, sort of in plain terms, some years ago, there was this phenomenal achievement that we all know, sequencing of the human genome. And I remember that we were all supposed to, stop getting horrendously bad illnesses shortly after this occurred. And it hasn't happened. Why? Okay, so there's the genome data, okay, and it's a very, discrete data. There's the phenotype that biologists have graciously been studying. I don't want to, I started out with biologists fixing the radio, making a little bit of fun of biologists, but we would not be modeling and simulation if they hadn't doing modeling and simulation or thinking about systems biology, if we weren't starting, on the, what do you call, on the shoulders of giants or whatever, on the shoulders of centuries of biologists making careful observations and learning about how things work. But there's observed phenotype, what organisms are doing, how they're reacting, and somewhere in the middle is this sort of vague network thing. Systems biology is about this network thing. What I'm going to be talking about is simulation of the network of biochemical reactions in a cell. Okay, and I'm going to be talking about discrete stochastic simulation. I used to, I still do, I have a fondness in my heart for solving systems of ordinary differential equations. And John Butcher can attest to that, wherever he is. Because we've, worked on this in that community for a very long time. And for the longest time, I thought ordinary differential equations, partial differential equations, the whole world could be described by these mathematical entities. Because after all, these are the objects that describe how things move. Okay? And I was just kind of frankly shocked when I found out that in cells, an ODE model or a PDE model isn't always valid. And why not? Because the action, F equals MA, or the action of ODEs and PDEs is really founded in the averaging of a great many events, molecular events. Okay, so I used to study computational combustion in my very first job after my PhD. And, in an engine, there are a great many molecules, and so to some, with maybe good approximation, you can, maybe not locally, in fact, but let's think more in a beaker. There are a great many molecules, and so they're all reacting with each other and doing things. And averaging all those actions makes a lot of sense. Okay, it makes a nice smooth behavior that's described by ODE or in space PDE. Okay, but a cell is very small. Okay, and in it are very large molecules. Okay, and in fact, something even maybe more surprising happens. Stochasticity can play a major role. Okay, in cells, in a real biological sense. Okay, in fact, to the extent that some molecules, which are present in very small numbers, are playing critical roles in a cell. Okay, and in fact, you know, it can lead cells to different phenotypes. Okay, and that's a good thing, evolutionarily. Okay, so for example, you know, DNA, you know, there's only one of those hanging out in the cell. Okay, so so an ODE model can't capture everything. Okay, on the other hand, a molecular dynamics model, okay, where we would model all the pieces of all the molecules with F equals ma moving around, is too slow, much too slow, given the model complexities and the time scales of interest. So what I'm going to be talking about here is discrete stochastic simulation. It's a kind of a mesoscale model. It's somewhere in between these two extremes. Okay, and why, the other part of the title of this talk, why spatially inhomogeneous? Okay, so in the past 7 years, maybe, I've been looking at spatially homogeneous models. Okay, those would be like the analog of ODEs. Okay, and that's those are founded on an assumption called well-mixed, that the chemical system is well-mixed, that spatial location isn't playing a big role. But cells are hardly well-mixed. Okay, and so of course you can think about, and we'll be doing that later in this talk, subdividing the cell into little volumes, each of which are well-mixed. That's going to be extremely computationally complex. Okay, there's going to be lots of fundamental problems with it. Just to show you a couple problems that we're working on in my group that have motivated such a strong transition in my group from well-mixed systems to spatially inhomogeneous systems. The first one has to do with unfolded protein response in an organelle called the endoplasmic reticulum. This is an organelle that surrounds the nucleus and is the gatekeeper for proteins going in and out of the nucleus. So it's a very important organelle. This is a confocal microscope view of two of the major players in proteins, BIP and Sec63, which is a chaperone and a co-chaperone. I'll tell you what those are later in the talk. You can see this. What you can see there is the sort of the outline of the endoplasmic reticulum. In there is the nucleus. Oh, why do I have this thing if I don't use it? There, nucleus. Here, endoplasmic reticulum. And you can also see that even along the membrane, these key molecules are not uniformly distributed. So we'd like to sort of understand what's going on here. Here's the other problem that's driving this research. I'd like to have some problems that are driving the research, right? Because they're always harder than the problems that you can think of yourself, that I can think of anyway. You know, biologists can always think of a harder computational problem than I can. Okay, so this has to do with polarization in yeast mating. So when yeasts mate, you probably don't lay up at night thinking about, you know, what happens when yeasts mate. But in any case, the yeast, think of it as kind of a, I won't try to draw here, kind of a blobby thing, right? And here's another yeast over here, okay, and they're emitting pheromones, tiny little gradients of pheromones that they're able to sense. They start orienting towards each other. They actually grow something called a polarizome. Okay, my friend Tomo Yi at UC Irvine does the experiments that make these pictures here, and he describes a polarizome as kind of the nose on the tip of the dog. I don't know why the dog has such a cute nose. It's really kind of an appendage, okay? In fact, they call it euphemistically the shmoo. I don't know how many people are old enough to know there's somebody who knows the Al Cap comic strip about shmoos, and it's this cute guy with this kind of appendage. It's actually reaching out to its neighbor yeast, looking to mate, and at the tip of it is this polarizome. Well, you might notice that down here, this guy has 3 polarizomes. That's because these are knockout yeasts. They're missing a key gene that helps it form only 1 polarizome. And we'd like to understand why these guys are forming multiple polarizomes and how they're being oriented, and how it's sensing the spatial gradients. Okay, so outline of this talk. The first part is going to be on discrete stochastic simulation for well-mixed systems, because you need to know some of these algorithms so that as a foundation for the second part. Okay, so I'm going to be talking about the chemical master equation, the equation that governs the evolution of the probability density functions of the system. Stochastic simulation algorithm, which is a kind of kinetic Monte Carlo method for that's specialized to chemically reacting systems. It could be very slow because it simulates every reaction event, every one. Think about that. And so there's been a lot of work in recent years on accelerating this in various ways, sometimes by rearranging the algorithm and using high-performance computing, sometimes via mathematics, which is the more powerful approach. And I'm going to talk about various accelerations because they all need to be used in order to tackle the spatially inhomogeneous systems. So I'll be talking about tau leaping, hybrid methods, slow-scale SSA, and a method called finite state projection. Then I'll move on to, if I'm still here and Ian hasn't kicked me off the stage yet, and to discrete stochastic simulation for spatially inhomogeneous systems. I'll talk about the version of the ISSA for spatially inhomogeneous systems. We call it the ISSA. Some fundamental issues which may surprise you. The use of some of the ideas from acceleration from up here in the spatial simulation, and complicated geometries, and then we'll get back to those applications. The second part is definitely work in progress. There's a lot of work to be done. Okay, so discrete stochastic simulation. This is where I started some 8 years ago. We start with a well-mixed system, okay, so no spatial dependence right now. We've got n molecular species, S1 through Sn. We're at a constant temperature. We're in a fixed volume omega. This part can be relaxed. We've got capital M reaction channels. That's what biologists call chemical reactions, R1 through Rm. We've got a dynamical state, X, okay, where X sub I of T, the ith component of X, is the number of species S sub I molecules in the system. So coming from differential equations world, this was a little bit strange to me, you know, that we're actually going to count numbers, integers of molecules in the system. But other than that, there's really no major strangeness here. Okay, the stochastic simulation algorithm was invented by, I'm really privileged to have learned all of this from Dan Gillespie, who was one of the giants in the field. He invented this algorithm in 1976. Just in case you're wondering, that's before I got my PhD. Mathematical audiences are the least funny. Nobody, in a math audience, nobody laughs at my jokes. And it's so bad that a few years ago I had already noticed this, and I was given a talk to the math department at the University of Michigan. And there was a big audience, and nobody was laughing at my jokes, except for one guy up in the top row, you know, and as a speaker, you find the one person who's paying attention. And you sort of key on them, because that can help you to give a better talk, right? And so I'm really keyed on this guy. I'm thinking, what's with him? He's unlike his colleagues, you know? But I'm happy, you know? And so afterwards, he comes up to me, you know, because fully engaged person from the audience comes up to talk to the speaker, and I say, who are you? And where are you from? He goes, oh, I'm a biologist from the University of Michigan. Okay. Okay, so anyway, these are some important terminology that you'll need for the rest of this talk. So Gillespie invented this in 1976. The same time the general KMC algorithm was invented. and completely independently. It's interesting that it wasn't very much appreciated in those days, and he had big arguments just in getting it published with the chemical physics community. And you know, I think timing is everything. Now he's a really mild-mannered man, and he's just shocked every time he goes into a biology lab, and the students kind of come with his book and ask him to autograph it and stuff. He's like, Linda, you can't believe what happened. They asked me to autograph their book. Who do they think I am? But anyways, so if you wait long enough, your great idea will eventually be recognized by the scientific community. Okay, so we've probably all had this problem, you know, where your best paper has the most trouble getting published. I always think that my best paper and my worst paper has the most trouble getting published, and I don't feel too bad about the worst one. But, okay, the SSA starts with this piece of vocabulary. The propensity function, aj of x dt, is the probability that given that we start in state x, that one Rj reaction will occur somewhere inside of that volume omega in the next infinitesimal time interval from t to t plus dt. And when that reaction occurs, it changes the state, okay, and the amount by which x of I changes is given by nu ij. Okay, for those of you who do ODE, chemical kinetics, reaction rate equations, you'll recognize this nu ij is the stoichiometric matrix. Okay, it's the very same one. It's the change in the number of S sub I molecules produced by one Rj reaction. Okay, so if I've got A plus B goes to C plus D, when I execute it once, I decrement the numbers of A's and B's, and I increment the numbers of C's and D's both by 1. And x of t is a jump Markov process. Okay, the stochastic simulation algorithm goes like this. It turns out that you can prove, Gillespie proved a long time ago, that the time to the next reaction is exponentially distributed. This is a key fundamental property of the chemical systems, and it's a foundation of a lot of this work that's often overlooked. Okay, so the first thing you do is you draw two independent samples, R1 and R2, from the set of uniform random numbers on 0 and 1. You take the first one, the first random number, down here it is down here, to define the time to the next reaction. A0 is the sum of the propensity functions. Okay, and you use the second random number, here it is, okay, via this inequality to determine which reaction is going to fire. So you've got the time to the next reaction and the one that's going to fire. You go ahead and virtually fire it, update the numbers of chemical species. Okay, so as you can imagine, there's a lot of reaction events that can fire in a real system. Okay, fast reactions, right? Species that are present in large numbers. Okay, so lots of people in recent years have been working on fast formulations of this algorithm. There's ways to rearrange it and make it more palatable on your computer or my computer or whatever. There's a next reaction method, the optimized direct method, sorting direct method, logarithmic direct method, and finally, if you're a computer scientist, you'll start recognizing a pattern here, right? We're O of n, O of log n, and constant time method, with the big constant in front of it, by the way. Okay, so we've, even in my group, worked on putting SSA on NVIDIA's graphics processing units, and it works tremendously well. The architecture is tremendously well suited to it. In spatial simulation, there's something called the next subvolume method, which is a rearrangement of the events that happen in the SSA. So, but there's only so much you can do with all of these approaches, because none of them is by definition able to reduce the number of reaction events. They're just all about executing them faster. And there's a great many reaction events. So here's where some mathematics can come in. Okay, and the first thing that happened, this was right before I met Gillespie, in fact, was he had this algorithm. In fact, I believe it's why I met him. because he had come up with this algorithm that he called tau leaping, and I've never been a fan of this name, but in any case, tau leaping. Here's the deal, okay? Instead of going the small length of time to the next reaction event, which is just, you know, a tiny, tiny little time step, what if we could do like in ODEs, okay? And at the time, I don't think Gillespie had ever heard of an ODE. He has ODE phobia when I met him, just like I had stochastic phobia when I met him. Okay, so, but the idea is, more like in ODEs, I want to take the time step of my choosing, not of nature's choosing, because I would choose a bigger time step. Wouldn't you? Okay, so suppose I chose a time step tau. Okay, then if we could determine how many times each reaction channel fired, okay, in the subinterval from now until tau from time from now, okay, then maybe we could forgo knowing the precise instances at which those firings took place. Okay, so I'm hoping I can just kind of go to tau and count up the number of times each reaction fired. Okay, I don't know the sequence of which they fired between now and tau, but okay, t plus tau, whatever. Okay, in that sense, we can leap from over a great many firings of reactions from one subinterval to the next. Now here's where I came in. How long can that subinterval be? Okay, so first of all, note that tau leaping, well, you don't know this yet, but it turns out that tau leaping is exact for constant propensity functions. Now, why does that make sense? Okay, because think about, if the propensity is constant, okay, then the order in which I fire reactions doesn't change any of the probabilities of the next reaction. Okay? If it's not constant, then every time I fire some reaction, it could change the probability of the next reaction firing, and then the order would matter. Okay, so it's exact for constant propensity functions, but real reactions, biomolecular reactions, don't have constant propensity function. Their propensity looks like a constant, okay, times the population of A times the population of B. Okay, so they're quadratic. Okay, so they're not constant. So what do we do when they're not constant? We rephrase the criterion. What else? We select tau so that no propensity function changes appreciably. Now, we and others have written a number of papers on how to do that. And I would say that at least for sort of non-stiff discrete stochastic simulation, I think we're doing that pretty well right now, but I don't have time to talk about it. Current implementations, oh, by the way, this is what tau leaping looks like, if you might wonder. Okay, it looks like xn plus 1, the population of species x at time n plus 1 is equal to xn plus nu, the stoichiometric matrix, times p. This is p of a tau is the Poisson, a random variable with parameters a and tau. It has mean and variance. It has mean and variance of a tau. Yes. I guess.
Okay.
Yeah.
These x's are still integers, right?
Yeah.
And so, how many of them are there? Because when you say appreciably doesn't change, and there were 2 of them.
Okay. That's a good question. That's a good question. If there's 2 of them, then we're not going to be using tau leaping. Okay, so for example, so let me go just a little bit over here. By the way, the Poisson random variable is the one that counts numbers of events, so it's not surprising that it's like that. But okay, so current implementations have adaptive step size, okay, and non-negativity preserving. Okay, so what happens, this is what's related to your question, what happens if you use tau leaping for situations where some species has such a small population like 0 or 1 or 2, which we certainly have examples of and they're important, it's not a happy situation to use tau leaping directly on that without modifying the algorithm. And why not? Because chances are it'll go negative. Okay? Now, not to detract from tau leaping, but 1978, that's when I got my PhD. I went direct to Sandia National Labs. I started solving combustion problems, and one of the very first things that happened was the ODE solvers were all going negative on combustion. So this has been happening before. This is not just tau leaping. But and I thought a lot about that. How can I just perturb my ODE solver? I know the solution to those chemical rate equations needs to be positive. There's even a parameter in my code that lets it do that in a dorky way. But you see, I use the word dorky and like nobody flinched. Amazing. Except maybe Australians don't know what it means. I can always hope. Okay. So anyway.
We just used to be called dogs.
Oh. Anyway. Okay, so it goes negative. What does it mean? It means that the approximation is not really valid. Okay, and that is true for tau leaping too. Okay, you cannot satisfy the leap condition if something goes from 0 molecules to 1 molecule, or from 1 molecule to 2 molecules. And what we do in an adaptive tau leaping method is that we have something called the critical species. Okay, and the critical species advances by SSA. If it has a population that's smaller than like 10, okay, it advances by SSA and not by tau leaping. The rest of the system is going by tau leaping. Okay, that's what this is about. Reverts to SSA when necessary. Okay, so hybrid methods. So, coming from the ODE world, okay, this has always been a dream of mine. How can we speed these things up? Okay, we've got some species that are present in small populations and others that are necessarily present in large populations. And large populations, well, we could probably get away with, this is a probably thing, okay? This is a hoping thing. It needs some mathematics behind it. But we could probably get away with simulating those with ODE, as ODEs, modeling them as ODEs, okay? Because they're present in large quantities, and we're going to average a lot of things. Okay, and these are called hybrid methods. This is just a small sampling of the number of papers. This last one, in fact, is for spatial simulation, a hybrid method. Here's how they go. Slow reactions involving species present in small numbers, okay, are simulated by SSA, just like we just discussed. If there's small numbers, simulated by SSA. Why not? If there's slow reactions, that's even better, right? Because then they won't slow things down too much because they don't fire very often. Okay. On the other hand, reactions where all constituents are present with large populations are simulated by reaction rate equations. Those are ODEs of chemical kinetics. Okay, so solving an ODE is blindingly fast compared to discrete stochastic simulation. So this sounds great. We thought it was great when we saw the first of these papers, Hazeltine and Rawlings. And then we tried it on our resident real problem, and it fell apart. And we found out very quickly why, because there's a hole in this thing, right? And what is it? What about a fast reaction involving species present in small numbers? At first we thought, well, maybe that can't occur. But sure enough, anything, in your nut years of numerical computations can occur. And in fact, it occurs in a lot of the examples that we've seen from real biological systems. Okay, so this algorithm cannot, and why would that be? So fast reactions, you can simulate them by an SSA. Fast reactions involving species present in small numbers. But if you do, it's going to slow down the whole business enormously, okay? Because they keep firing their fast reactions, okay? So we thought about that, and I actually think this is probably our very best achievement. And we came up with something called the slow scale SSA, or SSA. And what it does is that it simulates those fast reactions, even those involving species present in very small numbers, using something that we call the stochastic partial equilibrium approximation. Okay, so this is a stochastic version of partial equilibrium, okay, which it turns out all the chemical engineers learn in freshman year. But instead of having reactions go to partial equilibrium, okay, they go to what we call a stochastic partial equilibrium. Namely, their distributions go to a partial equilibrium, not the actual numbers of species themselves. Okay, so finally, usually people start with this, but I moved it to this part of the talk for what I think is a good reason. All what we've been discussing are approximations to the chemical master equation. The CME describes the evolution of the probability density vector, okay, for the system. Here it is. It tells all. The probability that we're in state X at time t, given that we started in state X naught at time t naught, okay, is given by this expression, okay, and what these terms here are, they represent if we started one reaction away from X and T, okay, these are the probabilities that each of those reaction J's would occur, okay, and this term has to do with if we happen to be unlucky enough to start at X and T, then these are the reactions that need not to fire, okay, for us to stay there. Okay, so the thing to notice about the chemical master equation, there's two things to notice about it. Number one, it's a linear system of ordinary differential equations. Number two, it is an extremely huge linear system of ordinary differential equations. Why? Because these are all the states. Okay, these are all the states. Let's say I had species A and B only. Okay, so extremely simple system. A goes to B, B goes back to A, something like this. Okay, so these would be all the states like I have 29 molecules of A and 5 molecules of B. Or I have 28 molecules of A and 6 molecules of B. Or I have 20, etc. Okay, imagine now I've got a real chemical system, okay, with all kinds of species, some of which are present in large numbers. Every possible permutation of numbers of molecules is a state in this gigantic system of ODE. And so the bottom line is that people don't usually try to solve this directly. But there has been some really interesting recent progress on doing some model reduction on this system, some very serious model reduction. Okay, and this is called, interestingly enough, it was invented by one of my colleagues at UCSB. A lot of work on it has gone on here in Australia. Kevin Burge has done a fair amount of work in this direction. And Roger Sidja? Okay. Anyway, so here's the idea of the finite state projection method. The CME, as I told you, is a large, possibly infinite, linear ODE. Okay, so we all know how to solve that. Okay, we just take the exponential of that gigantic matrix, and then we're done. Let those linear algebra people figure it out. Okay, so here's the idea. Obviously, you've got to reduce this model. Okay, so how is it done? It's modified. Kamash is a control theorist. Okay, so he how should I say? Somehow. Forget that I said that. Okay, anyway, he uses a truncated space. Okay, so only some states, remember I said there were a huge number of states, okay, that would make the system much too large to do anything with. What if we only kept the important states? Okay, and so this, of course, is the hard part, is to figure out which states to keep, okay, and which states to throw away, and there's algorithms for that. And when we throw away the states, we're gonna approximate all the thrown away states with what's called an absorbing state. Namely, if a reaction occurs that puts something into one of those thrown away states, it just stays there. Okay? Once you've done that, you have a smaller system. It may still not be small enough to be tractable, but if there's a lot of approximations that you can make, a lot of ongoing work here. Then you can solve it directly. Take the exponential of this now smaller matrix. Okay. The nice thing, a another nice thing about this algorithm is that the presence of that absorbing state provides a bound on the error. Basically, it's the amount of this exponential that we're neglecting. So I want to move on to spatially inhomogeneous systems. The base algorithm, we call it the Inhomogeneous SSA. The idea is to introduce a discretization of the domain into subvolumes or voxels, okay, just like we do for partial differential equations. We assume that the well-stirred assumption is fulfilled within each subvolume. That's being assumed in your partial differential equation too, by the way. And here's your subvolume. The diffusion is introduced as jumps from one subvolume to adjacent subvolumes. Okay, so this algorithm treats diffusion much the same as it treats reactions. There are probability for diffusion events, for molecules to diffuse, to transfer from one subvolume to its neighbors. Okay, on a Cartesian uniform mesh, the propensity for diffusion is given by a diffusion constant for that type of molecule divided by the spatial grid size squared. Okay. There are some fundamental issues that may be a little bit surprising at first with this, with doing reaction diffusion equations. In particular, this is pretty disturbing, I think. The limit, you'd like to take the limit h goes to 0. I like to take h goes to 0 and delta t goes to 0. But it is not attainable for physical reasons. There are conditions on the mesh parameter. Okay, this one over here assures that the system is well mixed, that the subvolumes are small enough that inside of each one we have a well mixed, and that's not so surprising. This one here, the lower bound on the spatial grid size. This rho r is a reaction radius. Okay, basically it says that I can't take the subvolume so small that the molecules don't have room to react. Okay, or else they won't. Okay, so these conditions were first pointed out in a paper by Elf and Greenberg in 2004. Sam Isaacson has done some nice work in this area, and he's shown that for reaction diffusion systems in the limit of small enough h with this algorithm, molecules never react. Okay, the subvolume just gets small and they only can diffuse, they can't react. There has been some theory and proposed improvements to the algorithm which amount to enforcing these kinds of conditions by Urban and Chapman very recently. Okay, now here's an interesting another strange thing that happens. Those reaction propensities vary with what's called molecular crowding. Okay, so a cell is a crowded environment. Okay, if it gets too crowded, and in fact the endoplasmic reticulum that we talked about earlier can get too crowded, then the propensities change as a function of that crowding. And in fact, they change in a differential way. Okay, namely, if you imagine you've got a crowded environment and there's all these big molecules hanging around, which molecules are going to be able to get around and react with other molecules? Okay, it's going to be the small molecules who can still move. The big molecules become orders of magnitude less mobile, their reaction rates go down, and it's actually being implicated. This is a really interesting paper down here about this happening in the endoplasmic reticulum of beta cells. Those are the diabetes, you know, when you're in the pancreas cells, and actually to an extent that the reaction pathway, the sequence of reactions that happen when proteins need to be refolded and the cell is in an unhealthy state, becomes different as a result of this crowding. Okay, so already for well-mixed chemical systems, we were looking at huge amounts of computer time for simulation. Okay, and I forgot to mention that usually you want to do actually you'd like to do about 1 million simulations of SSA in order to get good approximations to the probability density functions. Okay, so that's a lot of simulations. Often we settle for 1,000 or 10,000 just because that's a lot of computer time. Okay, but now think about putting it. This is the transition that years ago we went from ODEs to PDEs, and you know how much more computer time on our old slow computers those PDEs were taking. It was, I remember, it was painful. And it still is, you know, if you do a big enough simulation. This is the same order of magnitude computational complexity shift here, okay? And we were already having trouble with well-mixed systems. So what are we going to do? We got to solve part of this problem here with computer architectures. And the big fast computers are parallel computers. Even the little fast computers, the clusters of GPUs, are massively parallel computers. Okay, so we're going to put this thing on a parallel computer, this spatial stochastic simulation. And immediately, once you start thinking about that, diffusion is often the fastest process in the system because molecules have to get together before they can react. Okay, and so you can imagine, you know, you're in these tiny little voxels. And you've got molecules diffusing in and out between, you know, your current voxel and its neighbors. And they're going back and forth, you know, randomly walking. Okay? Well, you don't want all those big, fast, random walkers walking from one processor to another on your computer. That's a disaster. And so we've worried a lot about speeding up the process of fast diffusive transfers in discrete stochastic simulation. Our first attempt on this is something called the multinomial simulation algorithm. I think last year. It's a kind of, we didn't derive it with tau leaping in mind, but in retrospect, it's a kind of a tau leaping that's been specifically adapted to diffusion. In that the propensities for the diffusive transfers are conditional. Okay, in other words, if my molecule started in this voxel and went to the right, that means it didn't go to the left. Okay, so I need to take account of that. And this, for that reason, by the construction of this algorithm, it's conservative. It conserves. It's not conservative as opposed to liberal. But anyways, Diffusion FSP. We haven't decided how to name this one. Our next attempt at this, that's this year, that's right now, we're writing this paper. It's called Diffusion FSP, DFSP. It's an application of finite state projection to diffusion. Now, I mean, think about that. At first, it seems almost impossible, right? I've got this, all these molecules diffusing. There's an incredible number of states distributed in space. It's order of magnitude bigger state space than the reaction-only problem. But if we just specialize the FSP to the diffusion, with this observation, the diffusion from the current voxel, okay, in all directions, is independent of the diffusion of the molecule starting in another voxel over to here or wherever it's going to go. Okay, so we can do all these diffusive transfers at once, okay, independently. And when we do that, we can also do it with a truncated state space. We've got exponentials, okay, which we can store if we're using a fixed time step. And so far, this algorithm is getting a speed up of the first one got a speed up of, it was more disappointing than I had hoped for, about a couple of times, 2 or 3 times. Over just the straightforward algorithm. This one is getting a speed up of at least 15 times and getting a tremendously accurate answer, so I'm feeling very good about this algorithm right now. That just means that it hasn't run into the problem that would make me feel worried. Okay, I think I just told you about that, and Ian's starting to get nervous because he told me at 5 minutes of he was going to do something. I don't know. He knows that I tend to go a little bit overtime. This FSP algorithm was motivated by the yeast mating problem. Again, this is with Tomo Yi at UC Irvine. He's got this beautiful, it's amazing what experimentalists can get these days. This is a wild-type yeast, an unmodified B cell. See the beautiful polarisomes that it's building. And here is this knockout yeast with multiple polarisomes. So we're trying to model that. Of course, you know, in PDEs nowadays we're used to simulating complicated geometries and unstructured meshes. There's been a fair amount of work on how to do discrete stochastic simulation under these conditions just recently. Started with Isaacson and Peskin. There's some nice work on by Engblom firm, Helander and Lutstedt. That came out this year. It uses the dual of the Delaunay triangulation to create its well-stirred subvolumes. It's an adaptive hybrid method. They built a code. This is, I think, the most impressive part of this work. They built an interface for discrete stochastic simulation into COMSOL multiphysics. COMSOL multiphysics is oh, they'll kill me. I'm a consultant for them. It's like MATLAB for PDEs. Okay, but anyway, it's this huge amount of infrastructure and draws beautiful pictures and creates meshes and does all those things that Jamie has software for, but I don't. What they've done is create an interface so that when we have a new algorithm for stochastic simulation, we can just plug it in. But I would say this algorithm, if it has any faults, is that it's currently limited in its ability to handle stochastic stiffness, which I haven't talked about. Okay. Yeah, I'm running into Ian's boundaries. So I'll try to go as fast as I can. Okay, I talked about protein interactions in the endoplasmic reticulum. This is one of our driving problems that we've been working on for about 4 years with experimentalists, Carissa Young at University of Delaware and her advisor, Ann Robinson. Endoplasmic reticulum is an organelle that surrounds the nucleus. It has an interior that's called the lumen. It has an extremely large, irregular, almost fractal membrane surface. And it's implicated in folding proteins and also in translocation, in moving them, proteins in and out between the cytosol and the nucleus. It's called the gatekeeper for proteins. Here's these pictures you saw already. BIP, the resident chaperone in the endoplasmic reticulum. This is the protein that folds proteins, okay, that chaperones them, that enables them to translocate. Sec63 is a co-chaperone, which is present in much less smaller quantities, and it enhances the effectiveness of BIP. BIP is in a lot of different places at once. It has a tremendous amount of functionality. So here's a merged image of where, to what extent are the BIP and the Sec63 co-localized. We've been trying to model this, and we used this software interface into COMSOL. Andreas Helander, who was a student of Per Lutstedt, was visiting my lab for the last few months. It was really fantastic for all of us because you know, we could combine their functionality for irregular grids with our functionality to do stiff stochastic simulation. In any case, these are our early simulations of the concentrations of total BIP on the membrane, left, and the lumen, right? That's the interior of the membrane surface. And as you can see, it's kind of looking like the experiment. So we are guardedly optimistic. Here's a kind of an artist rendition of the ER geometry and how bizarre it can get. And this doesn't even illustrate all the bizarre things that can happen. And finally, I'd like to thank my collaborators. Anybody who's up there in yellow played a big role in the work that was discussed. My students and former students and postdocs. Here's my group as of last fall. We take a picture once a year. And I'd like to thank my sponsors, NSF, Department of Energy, National Institute of Health, and the U.S. Army. Thank you.